Thank you. And I'm very happy to be here and to give a talk on the butterfly issues. And the title is a model for selection of eyespots on butterfly wings. This work was done by collaboration with Dr. Ben Karaman and Untitled Chapter. So this picture that show you a rice field near my house, and very nice green, relaxed scene here. So just we could easily find around this field here, and this kind of butterfly. And the butterfly is somewhat dark, not so easy to find out. But if we could find the butterfly and look closer and closer, we could find a very beautiful eye spot pattern in the small creature, very, very small butterfly. So my title is related on this eye spot pattern. So first of all, I like to very fundamental of this butterfly. So as an observational fact, the first one is a focus point. This is a white, you can see this white spotted portion of eye spot pattern here. Focus point, it's a center of eye spot. The center of eye spot. The center of eye spot. And it is formed in the middle of a wing cell. Wing cell, not a real cell, but a small area surrounded by wing vein here, adjacent wing vein here. This is a wing cell we call. So the eye spot, and this one is located in the middle of a wing cell. Okay. And you can see while a certain wing cell could develop a focus point, this is a focus point, and in some wing cell, but other wing cells do not. And if we look at the wing, total wing, this is called a global pattern, and little is known about the mechanism that determine the global pattern element. But even though they are sometimes used for species identification, no? So our objective here is to propose a model for formation mechanism of global pattern elements, such as the number or locations of focus point on the entire wing surface, Okay. And before going into our model, I like to talk about some basic features of the color pattern formation. The question one is, what is color pattern on butterfly wing? The answer is color pattern. This is a very popular butterfly, Papiliozusath in Japan, is very popular. So an adult wing here, we look at closer and closer. These pictures are to be seen. And finally, color pattern is a kind of mosaic pattern of dozens to hundreds of fine tiled monochromatic scale cells here. Okay. In addition, I should say this scale cell, each scale cell is a cell and arranged parallel arranged from the proximal to distal direction for all the lepidopteran wings we could see. But the problem of the scale cell arrangement pattern is not on today's topics. And the question 2, when does color pattern develop in the life history? Okay, you can see this is a development, the process of due to yellow-colored butterfly, papillomemnon, a macaron here, here, and larval stage to develop, and pupal stage, and finally becoming the adult butterfly. So, and wing disc inside this pupal stage develop to be a adult wing. And we call this in the wing disc here. Oh, sorry. We can find that in larval and early pupal stage, and in which the vein system is simpler than that of adult butterfly here. So very simple. And also, this is a kind of cutted wing disc, transparent. Nothing we could see from this transparent wing discs. However, we call the pre-pattern without color, transparent, is produced in the wing disc, transparent wing disc in larval, the early pupal stage of development. Here is the original transparent wing disc, but if we stain by using the antibody staining technique, this is a picture taken in the wing disc of this butterfly, small spotted. This is corresponding that completely to the eye spot focus point. And further, some antibody technique revealed that in addition to the focus point, concentric color pattern already exists in this transparent wing disc in the larval or early pupal stages. Okay, this is the adult color pattern. Color pattern colors are just appeared just before the 1 or 2 days before emergence. Before that occasion, and nothing could be seen, just our eyes. So it is known in general the pre-pattern in the disk is the original feature of the wing color pattern. So color pattern formation process starts in the larval wing disk. So we therefore assume program of color pattern formation of butterfly wing is essentially that of pre-pattern formation in the wing disk. Okay. And the next issue I have to explain is two different kinds of research on butterfly wings. The first one is local pattern, and the second one global pattern. Okay? A local pattern means localized pattern, such as one eyespot pattern. This is a local pattern, and heart mark pattern is a kind of local pattern. This is also a local pattern. On the other hand, global pattern, the whole pattern covering the entire wing surface. And it's sometimes used for, as I said before, identification of the butterfly species. Okay? So, and local patterns such as an eye spot have been well studied, including genetic research. Some specific genes already discovered and identified, but little is known about the mechanism determining global pattern elements, such as the number of eye spot or precise locations of eye spot on the entire wing surface. Okay. As far as I know, nobody knows about the mechanism, I think. Okay. So I somewhat reviewed the research or analysis of eye spot formation. This is a local pattern, I think. The eye spot pattern formation consists of 3 developmental stages during larval and pupal stages. Okay? The first stage, establishment of eye spot focus point, from which, this is called the signaling center in the wing disc during larval stage. Here we can see the small white spot here. This is a focus point. And the second stage, activation and spread of focal signals through diffusion and concentric eye spot pattern determination in the wing disc during late larval and early pupal stage by gene expression, such as distal rest, engrade, etc., known to work. And the third stage, this is pigmentation gene regulation occurs. This is the color differentiation in the wing disc, just occurred a few days before emergence of adult butterfly this gene is also known these days, DDC, CNVA, GTPCH1, and etc. So it is well known there are 3 developmental stages. Our most interest issue is the first stage and establishment of isot focus point, especially entire global pattern element, entire wing surface. Okay? This is a copy of Carroll's book. The first stage is just a focus point determination, where the focus point appears in the wing cell. And then the next stage, second stage, is from the focus point, some chemical diffuse surround and to make a concentric pattern. This is another interesting story. And finally, based on this concentric pattern, pigmentation occurs just before a few days ago, few days of emergence. Okay, so we just focus on this first stage, not the second, the third stage. So there is some theoretical work, and one of which is done by Fred Nyholm, and this is done Fred Nyholm, the Duke, and he used the activation inhibition model here, and equation taking a certain appropriate parameter values here, you can solve two-dimensional rectangular space here. This number indicates the time series here. So he could obtain this kind of time series, and to get a stable pattern. The red point is completely correspond to the focus point in real butterfly. So, and in his numerical simulations, the vein here, oh sorry. This is a vein, a cell-associated wing vein. Vein acts as source of one of diffusing reactant. And okay, but this is a simulation of a local pattern, just one within one wing cell.
So the picture is that the activator?
Yeah, activator concentration. Okay. So I have to talk about another issues. This is called ground plan, and from which all other patterns are regenerated. This is not a real pattern, but a hypothetical original pattern, ground plan, we call. And major factor for diversification of global wing pattern is sketched here. And to understand that diversification, first the pattern is a band. Here, this is not a band, but originally a band, just right here. And B is disruption. A is symmetry system. B, disruption to divide into a small area by veins, okay? And C is dislocation, and just a slide along to the veins to make the pattern is very complicated. Slide here. And finally, D is expansion and expand and shrinking, finally elimination, and so on of pattern element. Okay, it is known this major factor is for diversification factor. Here. Our problem here is just after disruption, related to disruption and shrinking and expansion, etc. Okay. So from now, I have to explain new result. This is given by Professor Fred Nyhow, and this process show you a formation process of global focus point pattern in the larval wing disc of the butterfly species Junonia coenia here, the adult butterfly here. And this butterfly habitat is located in North Carolina. You know? Very popular, I think. Duke is in Durham, North Carolina. So just he took and breed and is doing some experiment by using this butterfly. So, and this number show you the development of eye spot development. Here, the one we couldn't any eye spot formation process here, but after time goes, and this one is probably pre-vein. Finally, this becomes a vein. And at the same time, from 4 to 5, something happened within wing cell here, here. And finally, we could find 2 candidates of eye spot focus, but left-hand side is disappeared and right-hand side just remained. And this one becoming to the eye spot focus point on the adult wing. So problem is why this eye spot developed, but this cell doesn't develop any eye spot. This is a problem, unsolved problem, I think. So our model is for selection of eye spot on butterfly wing. Okay. The basic idea and assumption here So basic idea is the key determinant of focus point selection is in the overall venation system of the entire wing disk. Right? The second, the key idea of our model is the changes in source value of the activator on the proximal boundary vane of the wing cell govern focus point selection. Okay? So we call proximal boundary condition dependent selection. And to analyze mathematically or computationally, we following hypothetical venation system where wing cells are rectangular and they are sources of the activator. This is already recognized on the authorized and Fred Nyhow and here. So as a first step, we assume this assumption. Okay. So not so good. Mathematical description of the model. This is a venation system of wing disk. Somewhat simpler. This one much simpler than the adult venation system. So two-dimensional wing disk with rectangular approximation. Here, this is a total wing. And here, or each one is a wing cell here. And gamma V is vane, ice vane, and gamma P, a proximal boundary here, proximal boundary. And sometimes this one is a marginal vane. This is the anterior-posterior direction. It's a proximal distal direction. Okay. And the mathematical description too is this proximal boundary condition here. This is a system. A1 is activator, A2 is inhibitor. So, and on vane, Vi and Vi plus or minus 1, and we fixed source values, A2 there. And okay. On the boundary, zero flux or two zero flux boundary conditions here. And reaction function here, it's Dirac-Meinhardt systems. And here, this one, we saw the positive steady state of ODE is given by this value. And the boundary condition at the veins, A tilde, here, like this.
Excuse me,
yeah.
Because now you have the notation, so the one where you have A tilde, which part of the boundary is that?
Here.
Gamma nu.
Gamma VI,
The
like
longer veins.
vein,
Yeah.
vein, longer, yeah,
Not
veins.
The proximal veins. The veins in
The
the
Here.
orange, the orange.
Orange.
These lines,
Orange
okay,
lines.
thank you,
Okay,
because that was hard to read.
and this one is just here, proximal boundary here, UX. Okay. The first one, the model with prescribed boundary condition, UX, and simulation, and the parameter value, and we adapted this and this kind of parameter values taken. And the first one, the constant proximal boundary condition. This is constant value here. From left to right, Kp is 0, 1, and 2. Here we could find, and here A1 SS is activated steady state value here. So some change. This one is one focus point is formed, and two focus points, and nothing occurs inside wing cells here. And other two cases simulated. One is concave shape, just like here, by using function, concave profile function here. And the convex function, boundary conditions, just like this. A concave case is eyespot focus points are formed as a steady state. But concave case, we couldn't find anything inside the cell. So this is a very important concave shaped profile produced eyespot focus point in the wing cell. Here, this one, this one just tests the sensitivity of behavior to geometry. And this is a function to generate this, not rectangular, but somewhat deformed geometry from a rectangular one. But the result are almost the same as the case of rectangular geometry here. Here, we show you some variation observed in nature from now. The first one is time series. This is a notch expression in Junonia coenia here. This is adult. We could see the process. This white arrow indicates a part of wing cell here. This one, and then the narrower, and finally some concentrated portion appeared in the wing cell, and in some case, finally this focus point moves toward the distal side here. And this is the final photos of this process. Here, we could reproduce by our model this directed movement from to distal direction here. This is final from here to here to a little bit migrate or I don't know migrate as movement or curves. So this is one example. The migration of point into distal direction is reproduced by numerical simulation by the model. We did not know about this experiment. This one sent from my colleague in Robert Reed. Okay, the next one, the second one, abnormal pattern resulting from incomplete vein development. This picture from my colleague in Japan. So just he kindly presented me to show, to be able to show in this occasion. So just is very not so easy to find. This, in this case, the sketch of this picture here. So just what happened is, and wing vein stopped to develop here, and then final eye spot, two eye spots appeared. So this is an abnormal case, and simulation by our model could reproduce this kind of two-spot pattern here. Okay.
So in this picture you have normal double eye spot, abnormal double eye spot which you show, but you also have normal.
This one is normal.
At the bottom, I mean the one at the bottom. In one cell you also have double eye spot there. This one, yes, this one.
Oh, this one, I don't know why this one two spotted. I don't know. But this one is, I think, another problem, I think.
Very interesting, yeah.
But this one, and if the vein stopped to develop here, so just the width of wing cell and extended, and as one wing cell here. So just this is somewhat wider, I think, compared with other wing cells. Okay. The next one is the third one is one eye spot split into two eye spots through addition of another additional vein. Here, this is a normal one, but additional vein added in here. So two eye spots appeared in their world. So just a sketch of this process, of this pattern is shown here. This is an additional vein. So numerical simulation could reproduce this splitting of two spots. Okay? So from now, this is our next version or next completed model, I think, a two-stage model for focus point selection and simulation. To generate in the previous talks, and just we prescribed proximal boundary profile, okay? But in this case, we generate proximal boundary profile by using this kind of equation systems automatically. So U1 and U2 parameter, just a variable U1 and U2 is two chemicals concentration, this activator and substrate. And the parameter values is here, and the gamma is taken to be a positive constant. This is called a scale parameter, I think. And the two-stage model is as follows. The first stage is about one-dimensional system is solved on the proximal boundary, gamma p. This is a union of proximal boundaries to steady state. And second stage, ux, this is a proximal boundary profile taken to be a function of the steady state obtained the first stage and weighted function here. So simulation of the second stage, the two-stage model, okay? The parameter value for the simulation like this, okay? The simulation 1 is 7 focus points on the ventral wingspan of an African butterfly Biciculus annana here. In this case, that U1, this is an activator, and U2 is substrate, and gamma is here, this parameter here. And in this case, gamma is the very low concentration level here. And in this case, all the 7 with totally 1, 2, 3, 7 wing cells we are using for computer simulation. Each wing cell develop focus point here. This is corresponding to this kind of pattern here. So the second example, no focus point on the wing here. There's no focus point. This is taken from the paper of Verdade 202. They made this wing by artificial breeding process, artificial hand pairing process. It took about 26 or 27 generations. And simulation is just here, and a little bit higher gamma concentration here. There's no eyespot on each wing cell.
Sorry.
Yeah.
So does this mean here you kind of your model also predicts the venation system, the veins, or?
At the vein.
I mean, before you always prescribe where the veins are. Right?
Yeah. The prescribed is just proximal boundaries.
Yeah, but
in this case,
vertical ones, these are veins. These can be interpreted as veins now.
The veins are in a fixed position. On the vertical veins, the concentration is fixed. It's a Dirichlet boundary condition with some prescribed function.
But that's the boundary conditions. But now these green lines in the middle.
That's the concentration profile.
Yes. So can these be interpreted as veins now, or
the veins are lines where you fix this So the veins for us are one-dimensional objects. They're
Given, yes.
which are given, and there's a Dirichlet boundary
Yeah,
condition. on the
but
vein.
now in
But
this
these
wing, it
interpret-
looks
exactly.
like if
That's-
there are
that's true.
structures, it
That's-
looks like.
That's very true. But that's where the locations of the veins are going to be.
Well, when you see those sort of fluorescence images, maybe the concentration profiles you see around the veins
Yeah.
could be compared with these. But I think the size of the vein relative to the wing cell it's very thin. So you
Yeah,
can-
that's
yeah.
true. That's true.
That
but that's not the point.
is not the question.
Yes,
The question is,
exactly.
does this
Yeah.
predict where
Exactly.
veins will
Yeah,
be?
yeah,
Or,
yeah.
Oh no, the vein
The
position is
position
given.
is given.
Is given.
We
it's
don't
prescribed.
predict. Yeah, yeah.
I mean, if you compare them with the first image where the is forming, you say that it is forming between two veins. So by comparing them to the first image, you would think that these are the veins.
These are the veins, yes, but we prescribe them. We don't. The model
Position
doesn't.
of the
Yeah.
veins is given.
It's given.
Is prescribed. It's not.
Yeah, but for why in fact in the other one you don't have the concentration around the vein? That is a question for me.
My question is, do these green structures which we see now kind of act as veins in the same way as the
Because
veins you
before
had
you
before?
don't have this.
They represent
along
veins.
the,
Yeah.
you
Yes.
don't have the fluorescence along the vein. Could you go before?
Okay.
I mean, one could also ask, what
I
are
am
veins
aware.
actually really? Yeah, so here it looks like.
These
The veins.
Yeah.
It's okay? It's okay.
It is exactly the same. Yeah.
The last one is a case study where the function gamma is dependent on the wing cell. Here is a blue line. Blue line indicates the gamma profile, just a stepwise profile. In this case, and lower value and the cells develop eye spot. And the contrary, at higher levels, does not develop the eye spot. Yes, this is we assume that this corresponds to this case, and this is adult 2 eye spot here, is corresponding to this. Yeah. So, and summary and discussion. We presented a model for selection of eye spot and butterfly. The first one is a model with prescribed boundary condition and simulation. The second one is the two-stage model as the next completed version and simulation. Simulation results could reproduce where phenomena occurs in nature and experiment. And the basic idea of the model are the key determinant of focus point selection is in the overall venation system of the entire wing disk. The second key idea of the model is that change in source value of the activator on the proximal boundary vein govern focus point selection. So we call proximal boundary condition dependent selection. Okay? But we are now asked to mainly two issues to extend the model with continuous 1D boundary simulation that could be changed in a mechanistic manner, just not a given and profile function, gamma function that automatically produce continuous. Yes, this is one. And the second one, to do more systematic analysis and quantification, including a robustness analysis done those in the following. This is the following slide, is just appendix. This shows you the influence of aspect ratio on eye spot force point determination here. The left-hand side, and boundary condition for the activator are taken to be proximal here. 0, left-hand side, right-hand side, twice the steady here, twice the steady variable. A variety of pattern or no pattern appears. So the current and future work. So just we are now asked extension of the two-stage model, so to able to generate the continuous proximal boundary condition profile. Okay? And second, systematic mathematical and computational analysis, including robotics analysis. And we are now collecting statistical data on distribution of eyespots on the entire wing surface. A lot of butterfly collecting to take data, and almost we have. So just it's analysis to combine the current model. And the fourth one is integration of both aspects. This is not just a mechanical or chemical aspect, but this is eyespot pattern formation and ecological aspect of eyespot, such as protection from predator and so on, to integrate two different aspects. Okay? So just finally, I acknowledge the Fred Nyhow and critical reading and various comment and suggestion. And Mr. Tokiwa of Amada and in Japan, Robert Reed, Cornell, for the permission to use the photo in our paper, and acknowledge the EPA SRC grant and TDE, and financial support of Lever Holm Trust Research Project. And this research started as Shekar was visiting Japan 2 years ago, and by supporting Japanese Society of Promotion Science Summer Fellow. Thank you.
Questions?
How do you know when the simulations have reached steady state?
Pardon?
How do you know that the simulations have reached steady state?
Tolerance for the time derivative.
But when the aspect ratio is large, you'd expect exponentially slow motion. So
It does take longer. Is the aspect ratio? I mean, it's. I imagine I'd have to check, but I think for larger aspect ratios, you had slower. It took a longer time for convergence to steady state. Yeah, I think that's right.
A related question, you always seem to assume or seem to observe that there's in any case a unique stable steady state. Is that, are there any examples where that's not the case? That you get some dynamic bifurcations, or
For the models that we picked, there's only, I think for the Schnakenberg, there's a unique steady state. You can prove that.
I think he means that. Do you have
for all these varying boundary conditions and so on, and varying geometries, you get varying state, you get varying
I see what we
variable steady state, so there must be
I
some
see.
kind of bifurcations.
In this
Yes.
parameter regime, I think, for these systems, you only have a Turing bifurcation. And you won't have oscillatory states, which would correspond to different types. I think
Yeah, but that's more difficult.
But we
because-
haven't,
Yeah.
in every case that we've looked at, you do converge to a steady state. But I think analytically one could show that for these two systems.
But there might also be multiple steady
Sure,
states, right, which
absolutely.
are stable and.
Absolutely. No,
I
we
mean,
haven't.
that's true, they definitely are, they certainly are. So you have non-uniqueness.
Yeah.
It depends on your initial conditions, I think. That's true.
Yes, did you study different initial conditions and see what you get? Because then you might get different behaviors.
Yeah, we've done a bit of that. So we've looked at perturbing the initial data by noise and looking at different initial it's
But
a
it seems
that-
you always do local near the homogeneous steady state.
That's in here, but you can also take initial conditions of 0. It's actually the boundary conditions, I think, which are giving you some robustness. But again, this is all numerical. There are no analytical results here. But you could take 0 initial data for both species, and you still see the same behavior.
I may have missed it, but is there evidence that this butterfly system actually is a reaction diffusion activator inhibitor system?
Yeah, that's originally introduced by Fred Nyhout a long time ago. And almost all people are trusted in the reaction diffusion system to generate the eyespot pattern.
Well, was it Nyhout's theory, or he found actual morphogens?
Exactly. I don't think it's not so clear to me. I don't know that there are morphogens that activate a inhibitor type that have been identified.
But Nyhout predicted this distal stripe. So he did the simulation first, and he said focus points formed by a stripe from the wing margin penetrating into the interior and leaving behind a spot. And before they saw that in experiments, that's what he'd been simulating. And then experiments saw exactly the same gene expression for DLL, I think.
Yes, but the question is, are
but
the molecules? Yes.
It may not be an activator inhibitor with a Turing system, but at least that was predictive in the sense that he saw it before they saw it in experiments.
What I'm thinking, and I run across this in the kind of work that I do all the time, is you have very similar PDEs which can give you the same pattern because they're all containing a Laplacian term, and if you have a geometry and some boundary conditions and a Laplacian term all in there, then some very different physical phenomena can give you the same mathematical function as your answer.
Is it that rough sort of thing that they're all somehow modeling short-range activation, long-range inhibition? They may not be, but there's a broad class of different systems that all essentially have that same principle behind them.
But the issue here, I think that's why Nyhout really proposed, is very keen on this, is that diversity is a big constraint. You're going to get these patterns by small changes in either genes or whatever, and that's the justification that you can, your constraint is really, you can reproduce these many patterns, but really by just small change, which could be associated to gene expression. And I think that's where, if you read this book, I think that's his main argument, that diversity acts as a constraint for the model, and it eliminates many of those models.
Which book?
Here's a book, I think,
Nyhout's book,
Oh, no.
yes,
Smithsonian
yeah,
Institution.
cannot remember the title. But there's
The big question is how does nature set up gradients biologically? I'm not convinced that it's always a reaction diffusion system.
Absolutely.
I mean, in that same direction, even if you know there are morphogens, what does diffusion really mean? Where does it come from? Is it some kind of signaling from cell to cell, which transfers the information?
At the moment, I think this is all hypothetical. I don't think there's any experimental evidence to support that there's an activator. You can identify an activator or inhibitor that's diffusing within, the wing disk. I don't know. Are there any experiments of this nature? Do you know?
Which feature do you like to know?
Well, the question, for instance, you model diffusion, so that is some transport. But morphogen is not taken out from one cell and transferred to the next, but it might be, there might be an activation process or something, right? Which is by some signaling. So, and maybe in some rough way this can be described by diffusion. I don't know. That was my question.
A completely different hypothesis might be that there's an interaction between growth. This is going on while the wing bud is enlarging by many fold. An interaction between growth and differentiation, which involves no diffusion at all.
At this stage, there's not a lot of growth, though. So the focus point formation happens when it's still in the cocoon, I think, when it's a wing disc, and it's quite stationary, it's quite static. The growth happens later, which is when there's disruption and dislocation and all these other processes. But actually forming the focus point, I think, happens on a
This is the
roughly-
first stage, I guess.
Yeah, this is which is
Yeah.
which is what's being modeled happens on a roughly stationary wing disk. It's not growing too much, I think.
Any other question? If not, let's
Okay, thank you.